Okay, so now we're really going to step up into the larger um, range of these polysaccharides. So now we're going to be dealing with definitely what we call macromolecules. These are going to be very large. And so there's a few of them we're going to be looking at here. So we got amylose, amylopectin, cellulose, and glycogen. So there's going to be four of them we talk about. And they're actually all made out of glucose. So it's the same monosaccharide, the same sugar, but we're just putting together a large number of them with different types of connections. We're putting them together differently. And so that gets us different sugars. It's kind of like the idea of an isomer where we have the same atoms and we put them together differently, we get a different molecule, but this is scaled up big time. And instead of atoms, we're talking about glucose. So amylose is going to be a series of alpha 1,4 glycosidic linkages. And I'm going to show pictures as I go through as well. And so this is what starch is. And so plants use this as energy storage. More often they use amylopectin, which I'll talk about in a little bit, but this is also something they'll do. And so it's just several hundred, 300 to 600 glucoses. And this is an alpha 1,4 connection right here. So I'm looking at my number one carbon. The O bond is going down, or the bond between the carbon and the O is pointing down. So that's got to be an alpha, and then it's glucose. So on the number 4 carbon, we go 1, 2, 3, 4. It's also pointing down. So don't let these thicker lines throw you off. This is still just a hexagon. We're not writing the hydrogens in here. This is just the copy I pulled off of Wikipedia. This is just their version, so it's free to use. It's a little bit different. So these are getting a little bit bigger. There's stuff I'm not going to be hand drawing in this video, so I'm doing screen capture. But this is what an alpha 1,4 linkage looks like. So we saw this with maltose, right? We just saw it between two. Well, if you do that several hundred more times, you have amylose. And so that's what amylose looks like. Amylopectin is very similar. I don't actually have a picture for this, but what you would have is every so often on the number 6 carbon here, because it goes 1, 2, 3, 4, 5, 6, this OH group would be used with an alpha 1, and then you make a branch. And you'll see it with glycogen as well, so that's why I don't have a picture. So plants can use amylopectin for energy storage as well, but the branching isn't very common. And so if you do see branching in this class, assume glycogen. I'll talk about it more when I get there. And so then next we have cellulose. This is used for structure in plants. This is what's used for cell walls, all that stuff. This is what makes plants rigid. This is very strong. And this is often how you see a beta 1,4 linkage drawn. So again, we just have glucose, and we go 1, 2, 3, 4, number 4 carbon. Bond goes down, goes to the oxygen, comes back, goes down. So there's not carbons here at the bends. Just be aware of that. So what would really happen is there's other ways of representing it that'd be a little more realistic. So it's not like the bend is going through, the bond is bending through space like this. It's just to draw it on paper easier. And this can go for several hundred, several thousand carbon or glucoses. It's a chain that just keeps going and going and going. And the interesting thing is, so we can break down amylose in our bodies for energy. We have enzymes that can break down these bonds, these alpha 1,4 linkages. We do not have that for beta 1,4 linkages. So that's why we can't do something like eat grass in our diet and get any sort of metabolic energy out of it. But we can eat sugar and get metabolic energy out of it. We can eat starches. Generally you cook them to make them taste a little bit better, right? You try to break them down a little bit through cooking. But we can break these glucoses apart using enzymes. We cannot break cellulose apart. We don't have enzymes for that. So things that do eat grass, like say cows, they can break cellulose down for energy, but they actually cheat a little bit. They have multiple stomachs, and in one of those stomachs is actually a microbe, a bacteria, that does the hard work for them. It actually breaks these linkages. That's why cows, they spit up, they chew, and then they swallow again. So sometimes they're bringing it into their second stomach where they absorb the loose sugars that the bacteria did not use themselves. And sometimes they're just sending it back to the bacteria because it needs to be digested by the bacteria more. And termites, they eat, wood structural buildings, they eat dead trees. they can break down cellulose for energy as well, but they actually do a very similar thing to the cows. They actually have a bacteria in them that does the heavy lifting. So they don't actually have their own digestive enzymes to break these linkages either. So it's very uncommon for these beta 1,4 linkages to be broken, and so they're not digestible by humans for sure. So how do we get energy out of these linkages? Well, what we do is we put a cow out in the pasture, we let it eat some grass, do its thing, and then we eat the cow. That's how we handle these linkages. That's what we do. We find something that can break them down, we let them do that, and then we eat the cow. Kind of like the cow is doing with the bacteria, except it keeps them alive. It doesn't eat the bacteria, it eats the sugar that they let loose. This is what plants use for structural reasons. Amylose would be what they use for energy storage. So very different purposes, but it's the same building block, which is pretty incredible, right? It's like how you can build a wall out of bricks, you can build a road out of bricks, you can build a bridge out of bricks. It's different functions, but you're using the same little individual piece to do it. You're just applying it differently. And plants are doing that with these sugars, with these glucoses. So what about animals? What do? Well, we use glycogen. And so you better believe we can break glycogen down for energy. And so glycogen is going to be a lot like amylopectin. So it's going to have those alpha 1,4 linkages making a long chain. And then every, 20 or so glucoses, you're going to have an alpha 1,6 linkage that causes a branch. So this is like amylopectin, but where amylopectin has infrequent branching, glycogen is going to have frequent branching. We have a lot of branching. I'll talk about why in a little bit. So this is energy storage in animals. And so what am I talking about with these linkages? Well, I've got my alpha 1,4s, and then I've got a alpha 1,6 linkage here. And then what happens is then we just have more alpha 1,4 linkages. It just carries out. It just keeps going. And then it can branch again and keep going. You branch down here and keep going. So you get a lot of branches. And so when we zoom out and look at it, this is what we see. There's usually a kind of a protein in the middle. That's what you're seeing here with the ribbon structure. If you're thinking that looks like a protein, like I'm seeing some alpha helices and stuff in there. Yeah. But that's just sort of, the center point, what's coming off of it. This is all sugars. And so we have a long chain of alpha 1,4 linkages, and then boom, we have a branch. We have an alpha 1,6. Over here, we've got a chain of alpha 1,4 linkages, alpha 1,6. And this is actually scaled down a little bit. They might get a little bit bigger. Now, we have a ton of branching, so it kind of makes like a blob. And so this is energy storage that we would use in our aerobic respiration process. We would break these down, we'd get the glucoses out, we'd burn the glucoses for energy. We'll talk about that process more later, but this is how we store that intermediate energy storage in our bodies. And so if you start to exercise, you'll actually start building up your glycogen stores, and that's why you're able to, among other reasons, everything starts tuning up, and this is one of them. When you start exercising and you start seeing improvement, maybe you can run farther or faster or lift more or whatever it is. Although lifting's more anaerobic, but you'll start to build up these energy stores for those purposes. So why do we have all this branching, and why don't plants? Well, we live very different lives from plants. If you didn't know that, you've now learned something taking my class. You're welcome. We are not plants. We do not have the same needs. So when a plant is storing energy, they basically just have to make it through the night, and maybe or winter, but it's a known amount of time. They're gonna break off these glucoses and burn them for energy at a pretty steady rate. The sun's out, they're gonna build. The sun goes down, they're gonna break it apart, and they just have to make it till the sun comes back out, and then they start building them up again, because they're getting energy from the sun, and then they're storing some of it in amylopectin. So there isn't a need for as much branching, because when it comes time to break it down, they don't have to break it down very fast. It's a pretty slow burn. But animals have variable energy needs. If I'm walking along, I'm using energy at a certain rate, but then I see a lion is about to chase me. Well, you better believe I'm going to use every last sugar in this thing to survive if I need to. So I'm going to burn it fast. I got to get away. Like, it's not like, oh, if the lion catches me, I can escape later. It's I have to escape now. So I have to, fight or flight, and I'm gonna flight. It's a big lion. I'm gone. And so what happens is we can get more sugar out because we have more branching. So every one of these endpoints is a spot where we can start breaking off sugars and burning them for energy. And so the more branching there is, the more sugar we can access immediately, the more energy we have immediate access to. Now what happens is I can run all these chains down. I can have an enzyme pop on, it just starts breaking off these sugars as needed. But then it gets to that 1,6 linkage, and another enzyme has to come in and break the 1,6 linkage, and then it can start breaking the 1,4s again. And so it can slow down at the branches. So for plants, there's no need to deal with that complication of having a bunch of branches, because they're not going to outrun a lion. In fact, the lion's no threat at all. What do plants have to worry about? Well, they just got to worry about making it until the sun comes up. If something comes along and starts eating them, there's really nothing they can do about it. They can't move away. And then the other big threat would be, well, another plant gets taller and starts shading them. So they've got to. That's how plants kill each other, by the way. They just throw shade. That's how plants kill each other. They just throw shade, and then the lower plants below them don't get enough sunlight, and they die. Like, they literally throw shade at a fatal level. And so they don't need to deal with these branching issues because either they're going to make it or they're not. And so really all they can do is just kind of hunker down for the night, and they don't worry about the other stuff. Whereas animals have these variable energy needs. Not only do I have to not get caught and become something's meal, but sometimes I have to catch something and make it my meal. That lion's doing the same thing. It's burning down energy. And either you catch something or it gets away. There's no catching it later necessarily. I mean, a little bit, but, in the terms of the chase in the moment, not so much. And so branching allows faster release of energy, but a little bit more complicated of a process. And so that's why animals have more branching, plants have less. So if you ever see this 1,6 linkage branch here, assume glycogen, because it's so frequent. Whereas in amylopectin, it would be every couple hundred. Glucoses, it's like every 20 or so in animals, so it's very frequent. And so that would be reflected in the pictures as well. So if you see a beta 1,4 linkage, you're like, oh, it's cellulose. If I see that little zigzag, cellulose. If I see it, something like this or like this, I know it's not cellulose. And if I see the branch, glycogen. If I don't see a branch, I just see a bunch of alpha 1,4 linkages, then it is amylose. And I just won't throw a picture of amylopectin your way. So there's really only 3 you have to know, but be aware that this does exist. And so these are the large macromolecules. So this is carbohydrates scaled up to the max. There are a few other things, like you can look up chitin if you want. That's what arthropods use as their shells. So like bugs and what else got, like lobsters, things like that. Crayfish. Their shells are actually very similar. There's some similarity to cellulose, but there's some other stuff mixed in as well. So they're very interesting. Feel free to look that up if you want, but not super important to, human medicine. So I'm not worried about it right now. But yeah, so that'll be it for this section. Hopefully it makes sense. So those linkages are very important for being able, if you can recognize the linkage, you're going to have a much easier time recognizing what, large polysaccharide you're looking at. So that idea didn't go away. And this was just for illustrative purposes on the large scale, because glycogen has a very interesting, macro scale here. But this is what we'd really be looking at is this ring structures and how are these ring structures connected. All right, so that'll do it for carbohydrates. We've got one more biomolecule to talk about after this. That's lipids. That'll be in the next section, and then that'll be it for biomolecules. So if you have any questions about carbohydrates, as always, you know the drill by now. Use the forum, email me, whatever. However you want to contact me or ask classmates for help, totally fine. Have at it. I have no issues answering questions. But this is what I expect you to know in terms of these large ones. So being able to recognize what monosaccharide you're looking at, whether it is fructose, glucose, galactose, or ribose, being able to put them together with known glycosidic linkages, or being able to label the glycosidic linkage between two monosaccharides. Those are all fair game. And then being able to recognize these really large polysaccharides that we just talked about in this video. That's what this section really breaks down to. So if you're struggling with any of that, I'm happy to help. Just let me know. Otherwise, have fun with lipids next.